Okay, so let's start the class. In the last class, what we were discussing was we were talking about the kingdom called as Monera, right? So as we have discussed, we have talked about the salient features or very important points about Monera. Which point is important from examination point of view? Monera boli kole, the first line that we remember, it is the kingdom of prokaryotes, right? It's the kingdom of prokaryotes, and talking about a prokaryote, the best example of a prokaryote that we study in details is obviously a bacteria, right? So in the last class, what we were doing was, since we know that Monera is divided into how many? 8 types of Monera, and out of that we were discussing the first type, that is eubacteria. First mane, we are not going in a sequence. Obviously, we have already written it about archaebacteria and the different forms of Monera. So eubacteria, we have started, we have drawn the structure of the prokaryotic cell or the structure of the bacterial cell. As we have discussed in the last class, what we said is, out of all the shapes of bacteria, which is the most common shape that is available, that is the rod shaped or bacilli, right? And so we have drawn the structure of a rod shaped bacteria. So prokaryotic cell, we are not going to draw it again since we have already drawn it in the last class. What we remember, that it has a cell envelope, and if you talk about the cell envelope, it consists of how many layers?
3 layers.
It consists of 3 layers. It's the cell wall, it's the capsule, the cell wall and the cell membrane. Inner to the cell membrane is the cytoplasm. In the cytoplasm, the genetic material that is called as nucleoid. The nucleoid is also described that, or it also has circular DNA, right? We have to remember that word. Histone proteins present or absent, histone proteins absent, right? Apart from that genetic material, extra genetic material that is called as plasmid, right? Plasmid was the extra genetic material found. Apart from them, ribosomes, certain types of granules, right? So these are the structures we have discussed, and plus together with that we have talked about a structure which helps in conducting cellular respiration in case of bacteria efficiently. So which structure am I talking about? It is the mesosomes, right? So mesosomes at a point monot rakhibo disilo je cellular respiration tat hoy and ATP production kot hoy mesosomes hoy. Next is 2 types of mesosomes, septal mesosome and the lateral mesosome. What is the function of the septal mesosome, to be precise? It helps in the formation of septa, or it helps during the process of cell division. So this was the content that we have discussed, and obviously about the hair-like structures that is found in the bacterial cell. 3 types of hair-like structures are found, that is the pili, the fimbriae and the flagella. Out of the 3 types, it is only the flagella that helps in locomotion. Pili aru fimbriae function ki asile? Pili aru fimbriae function ki? Pili aru fimbriae ki hoy? Hair-like structures, no? Corpora ulai. Capsule বা outer layer পৰা ওলাই function to কি আছিলে attachment, right? So 3টা hair like last class diagram to মনত আছে নাই আছে ন? So hair like structures আছিলে, তাৰে flagella টো হে অকল locomotionত help কৰে আৰু বাকি কিটা কি কৰে attachment বা লগ লাগি থাকিবলৈ help কৰে. Okay, so we are going to continue that. আজি লিখা cell envelope. And those students who has already written it from the PDF, you don't have to write it. Cover audio problem hoise mic to mute kori loa. So as we have discussed, cell envelope or first layer ki capsule, and what we remember is that not necessarily all the bacteria will have a capsule. If the capsule is absent, they have a layer that is called as a slime layer, slimy in nature, and so it is called as a slime layer, right. Slime layer is made up of certain type of sugars, and in the capsule also you see that it's made up of polysaccharides and D-glutamic acid, right. Generally ekhini portion iman besi important nohoy, kintu ketiaba hudhi dibo pare je dextran, dextrin or levin is present in which layer, or it is made slime layer is made up of which, but not a very frequently asked question. Next is, if suppose I think last class we have discussed eta bacteria capsule thakile ki hobo aru nathakile ki hobo, right. If the capsule is present, then it will make the cell virulent in nature. What is virulent? Disease causing property. If it has, then it's called as virulent, right. Aru jodi capsule to nathake tete hole disease causing capacity to nathake, and plus it could be killed easily, right. So these are the points. Next, slime layer function ki likhiso, it protects the cell against desiccation and loss of nutrients. Loss of nutrients we understand. What is the meaning of desiccation? Drying up, right. So that the cell does not dry up, that is what is called as desiccation. So water content to maintain koribole kune help koribo, it is always the slime layer, and loss of nutrients. Please write this part. So this is all about the cell wall, the capsule. Basic monon narakiba, 2 points that you remember: capsule might be present, might be absent. If present, it makes the bacterial cell virulent. If this is over, next layer ki hobo dekhason cell membrane. Cell wall to olop pisot korim, cell membrane likhiba. Made up of cell wall is complex. Likha se di likha monot thakilei hol hoy ne, mane we just use a word that is complex. Complex not necessary. After the end of the lesson you will have an idea what are the points that you need to remember, right. Karon bacterial cell wall boli kole we always remember one thing: peptidoglycan, right? So next to the cell envelope, next to the capsule was the cell wall. But cell wall we will discuss later. Prothomote cell envelope plus cell membrane likhason. Is this part over? Online? Okay. Thik ase. Baki khini likha hoisile nai? Okay. Okay. So the next layer, we are talking about the innermost layer. Innermost layer is called as plasma membrane, or it is also called as cell membrane. Talking about the plasma membrane, the characteristic point is present in each and every cell. Either it's a prokaryote or it's a eukaryote, either it's an animal cell or it's a plant cell. So both plasma membrane is something that is going to be present. As far as the structure of the plasma membrane is concerned, how does the plasma membrane look like, or is it made up of what? Do you have any idea? Plasma membrane to kenukwa dekhat ba plasma membrane boli jodi hudho, the chief constituents of the plasma membrane: lipids and proteins, right? I think you all have read, no? We have not discussed, but you all have already studied the chapter called as cell unit of life. Cell unit of life or plasma membrane is something that you have to study in details. And as far as the structure of the plasma membrane is concerned, how does it look like? Various scientists from time to time has given different models. Remember something called as the sandwich model? Yes, the sandwich model, then the trilamellar model. What is the most accepted model that describes the structure of the plasma membrane? Fluid mosaic model, right? Singer and Nicholson. So tahate ki disile eta specific model disile, and that model talks about how does the plasma membrane look like. What did they say? That they said that the plasma membrane is semi-fluidic in nature. Agor scientists bilake, what they thought that the plasma membrane is a rigid structure, but it is Singer and Nicholson who said that it is not a rigid structure, instead it is a semi-fluidic in nature, means it's not solid, neither it's completely liquid like water, it is fluidic in nature, right. Next point that they have said is it is made up of phospholipid and protein, right. Phospholipid or keita layer thake, protein or keita layer thake? Phospholipid or duta layer thake. Okay. So since we know that the plasma membrane is present in each and every cell, so obviously the structure of the plasma membrane in case of a prokaryote and the structure of the plasma membrane in case of a eukaryote always will be the same, right. So how does it look like? It almost looks something like this. Ebor ki hoy tadpole flagella thakile amar enuka lage no tadpole jen. Okay. Okay, we are not drawing in details, neither we are marking each and every part, because this is something that we are going to discuss during our cell chapter, right. As of now we know if you are talking about the plasma membrane, it looks something like this. It is made up of what? It is made up of lipids and proteins, right. Talking about lipids or phospholipid, it has two layers, so we use the word bilayered or two-layered phospholipid. Next is, together with the lipid we have different types of proteins also. If you remember from that chapter, tinta type of proteins present asile. What are the three types? Intrinsic, extrinsic and transmembrane protein, right. There are certain types of proteins that are found just above the membrane, and those proteins are called as extrinsic protein, right. There are certain proteins that are embedded inside the membrane, and that is what is called as the intrinsic. And obviously we have the transmembrane protein that helps in the process of what? Transmembrane protein bilake ki hot help kore? FD, facilitated diffusion, right. So we also have, or we also use a word called as carrier proteins, right. Carrier proteins bilaka kam bilak kune kore? Transmembrane. What they do? They carry the substances from one cell to the inner contents of the other cell. Another point that is important is, whenever we talk about the plasma membrane of a prokaryote, mesosome present. Ebilak ki hoy? Ebilak pisat cellot ami korimi. Head or tail details ami cellot poribo laibo. If you remember, you have studied something called as hydrophilic part and the hydrophobic part of a phospholipid molecule, right. So hydrophilic head and the hydrophobic tail, right. So details ami hetu chapter out korim. As of now, this is all about the cell membrane. If this is over, please write about the cell wall. Can I rub the board? Online students, musibo paru niki? Okay. So the next layer would be likha cell wall. That first point ki likhiba? It is a layer in between the capsule and the plasma membrane. Let me write the points and then we'll explain. So the basic points, it's a layer in between the capsule and the cell membrane. We understand it is semi-rigid structure. What is the function? It prevents the cell from expanding and bursting. So this is just a common sentences that we write about the cell wall. Iman questions here pra ahan hai. Now talking about the cell wall of bacteria, whenever we talk about bacteria, I think you all have heard something called as gram-positive and gram-negative bacteria, right? What is the basic difference between a gram-positive and a gram-negative? Kuntu bacteria kami gram positive বুলি কম আৰু কোনটো ব্যাকটেৰিয়া আমি gram negative বুলি কম actually whatever you answer I don't hear yeah on the basis of what bacteria has been divided into two types actually classification of bacteria to bahut types of ase no kintu on the basis of a particular property they have been divided into either it's a gram positive or it's a gram negative on the basis of which property it's the beginning of staining and which stain are we talking about gram stain right so it's called as gram positive or gram negative we'll come to the staining part later so since we are talking about that two type of bacteria no some bacteria that retains the color some bacteria that do not retain the color the next question is why is it like this that some bacteria takes up the color and some bacteria do not retain the color gram positive kisuman colorful hoy gram negative colorful nohoy what is the reason behind the reason is it is because of the structure of the cell wall right it is the cell wall the structure of the cell wall of gram positive so gram positive is a bacteria gram negative is also a bacteria so obviously they will have a cell wall right talking about the cell wall the formation the structure of the cell wall is slightly different that is one reason why we can classify them against gram positive and gram negative now we'll just have a rough idea nothing in detail say gram positive or gram negative or cell wall too what is the basic difference right okay talking about the cell wall of bacteria the first word that we remember is obviously peptidoglycan jeneke cell wall of plants ba plants or cell wall boli kole it is made up of ki cellulose jeneke fungi we remember it chitin or chitin right so similarly if you are talking about bacteria the first word that comes to our mind is peptidoglycan so obviously peptidoglycan would be present here it would be present here also but the amount of peptidoglycan in both the cases would vary yeah please come So a question has never been asked as to identify the cell wall of gram positive and gram negative. The point that we are discussing here is just to give you an idea that there is a difference between the structure of the cell wall of gram positive and that of a gram negative. What is the basic point of difference that you can see in both the diagrams? It is the amount of peptidoglycan. Peptidoglycan, as I said, obviously will be present in both the cases, but the amount would vary. So in case of gram positive, it is more, it is obviously thicker as compared to the gram negative cell wall. Peptidoglycan kom thake. Next, another point of difference is about the cell membrane or the layer, right? So in case of a gram positive, you see a single layered structure of the cell membrane. Instead, in case of this, you see a layer that is above the peptidoglycan also. So duta layer thake, eta covering layer thake. So gram negative bacteria, what it has is, it has an extra covering that is made up of peptidoglycan, which is absent in case of gram positive. So yate okol eta layer phospholipid or yate duta layer, right? So because of these certain changes, no, that is the reason, maybe because of that they do not take up the stain. It will be very wrong if I say that they do not take up the stain. The correct statement would be they will not retain the stain. Take the stain and retain the stain, duita kintu beleg words hoy, right? Okay. Next is one point that is very important is thichoic acid. Okay, thichoic acid obviously does not look like a thiconi. It's just a way to represent so that you understand, right? Thichoic acid no, it's just a way so that you all pay attention to the diagram and you remember that there is something called as a thichoic acid, right? So in case of a gram positive, we will be talking about differences between gram positive and gram negative. So question or bithor, but difference or bithor, the presence of thichoic acid is very important. Question ahe, thichoic acid is a characteristic of gram positive ne gram negative? It is a characteristic of gram positive. Right. Second point, what is the function of thichoic acid? So all these functions are properly written in your PDF. Since you have already got the PDF, as far as the writing portion is concerned, you can go home and write it down, right? Here what we will do, thichoic acid or information right? Because that's not very important also. The first line that is important is, it is a chief characteristic of a gram positive cell wall. What is the function of thichoic acid? It acts as a binding site. Do we know what is the meaning of binding site? Okay, heard about bacteriophage, right? Bacteriophage of ami ki dekhisilu je virus eta, the virus get attached to what? It gets attached to bacteria. Bacteria, if you talk about, it's a big cell, right? Big mane no comparatively whatever it might be the size. Now the point is, virus kuntu part of attachable. That is decided by the thichoic acid. In which area the virus will come and get attached with the bacteria, that area, that point of binding is something that is decided by the thichoic acid, right? So what is the main function of thichoic acid? It acts as a binding site or it acts as a receptor, right? But that is not important. Important is, thichoic acid is a chief characteristic of which type of a cell wall? It's a chief characteristic of a gram positive cell wall, right? Actually, sabole kole cell membrane boli. So basically, cell wall made up of peptidoglycan, and inner to the cell wall, it should be the plasma membrane or the cell membrane. So this diagram is according to that we have discussed. But in this case, what we have seen is, obviously we have seen the cell wall, we have seen the cell membrane, but on top of the cell wall also, we will see a phospholipid bilayer. Since it is phospholipid bilayer, so we are using the word called as cell membrane. Actually, we should be using a word called as an extra covering over the cell wall, right? Oh, eta word monot rakhi, bacterial cell wall, peptidoglycan. Polysaccharide is an example of a polysaccharide, right? Polysaccharide, so it will depend on the option. Option ki ase. In maximum of the options, peptidoglycan e thakibo. Jeneke cell wall of plant cell boli kole cellulose e thakibo, no? Thik teneke bacterial cell wall chief component is always peptidoglycan, right? And we'll come into details. We'll have to talk about peptide part is made up of what and glycan part is made up of what. Certain types of amino acids, no? Peptide part or to kisman amino acid thakibo, glycan part or to. Iman important no hoy. But we will not miss because keteba hudi hudi bo pare. Important kuntu point we have already said. Remember the word peptidoglycan. Okay. Next is, we have also written something here. What does this line tells us? It talks about staining, no? So jedi gram positive hoy, they will take up the stain, they will retain the stain, and they will obviously become bluish or purplish in color because of crystal violet. And if it is not gram positive, then পাৰ্পল কালাৰটো নলয় পেলাই কি হৈ যায় কিয় পিংক হয় Okay, before going to that color or staining, আমি peptidoglycanৰ অকমান information লিখি লওঁ. Is this diagram over? Okay, অকমান extra point লিখা peptidoglycan বুলি heading কৰা.
No doubt we have written a lot of information again. So when we are writing a note or when we are discussing in a class, obviously we say certain things extra, right? But you have to be intelligent enough as to what you will read for the exam, right? So after every class or in every class, moi kisuman points stick kori diu, no? Every point I say what you have to read and what you don't have to read. As far as preparing the note is concerned, note to gutei likhibo lagibo, because you never know what is going to be asked in the next examination, right? So previous experience or we have an idea of what is the question asked about or asked from this portion. right? So we have written so many things. What we know, the first point, talking about a bacterial cell wall, it is always made up of peptidoglycan, right? So peptidoglycan is made up of what? Peptidoglycan is made up of certain types of amino acids, right? What are the different types of amino acids? Questions generally aha nai. Kintu, there are 2 points that are important which I have marked, right? 1, 2 and 3. Right? So if you see, the peptide part is made up of 4 amino acids and the glucan part is made up of 2 amino acids, right? Out of these 4 amino acids, what is the point that is important is the type that is present only in case of gram negative. All of what we have said is, we have talked about a characteristic point of gram positive cell wall. What was the characteristic point? The presence of thichoic acid. Another important point, now we will be discussing about a main point about the gram negative cell wall. Gram negative cell wall or what's the important point ki ase? DEPA. So question is directly asked on that. DEPA is a characteristic of gram positive ne gram negative. Thichoic acid is a characteristic of gram positive ne gram negative. And as far as these amino acids are concerned, I don't think you need to remember. Karon questions aha nai tenike. Thik ase? Khali ketiaba sudibo pore L lysine is a characteristic of what? And DEPA. If you remember DEPA, then obviously we know that lysine is not a characteristic of in case of gram negative, right? So that's a point that's important. Next is coming to the glucan part. Glucan part is made up of alternating units of amino acid. Alternating, I think we understand. Ebar jodi neg thakibo, next time you'll have nem. If the next time, if it's nem, the next time you'll have neg, and so it will be in a alternating sequence, right? Neg and nem, it's important. Why it's important? Because we will be talking about different types of Monera. It's not only eubacteria that we will be discussing in the class, right? Eubacteria is a Monera and some other bacteria, for example, Archaebacteria is also a Monera, right? So basically if they are Monera, their cell wall will also have certain points of similarities and certain points of differences. So we will be comparing between 2 types of bacteria, eubacteria and Archaebacteria. One point of difference is something about this. So when we will be discussing about Archaebacteria, at that time we will be talking about this again, right? So question to ahe, Archaebacteria ki thake ba ki na thake. Out of this, no, kiba eta thakibo, kiba eta na thake. Instead of that, there will be something else that is present. So let's not go into that. As of now, neg and nem, full form, iman important no hoy kina neg nem jehetu porhibai, N-acetyl glucosamine or N-acetyl muramic acid, you need to remember. So this is all about the cell envelope. So last class we have drawn the diagram. We have discussed certain important points about the parts of the diagram, and today we have completed the cell envelope. So as far as writing is concerned, we have written a lot of information, but if you remember, I have asked you all to write certain types of short notes, no? First class ote koisile ne short notes likhibole. So what you do, class of bitor to bahut likhila no, ঘৰত গৈ পেলাই short note যোনগিটা point tick কৰি দিছো ন হেগিটা লিখি থবা last moment অকল হেগিটা চালে হ'ব develop the habit of writing a short note. You don't have to write by heart each and every word that you have written in the class, right? Only the tick point or maybe the underline point you can write it extra and paste the paper in your copy itself, right? So the next that we are going to study is normally staining in bacteria boli kole we have an idea of what we are talking about, right? So we don't need to write everything, but in spite of that I will give the flow chart. How do the process work? Ami first dot ki koru, second dot ki koru, stain kenege koru. The reason behind giving the flow chart is how. Yes my dear, last bench, what happened? Kiba hoise ni buji na nai poa neki kiba hudisila kotha patisilo কিবা এটা অপশন টু কারেক্ট হবই ন এমসিকিউ যেহেতু ওকে সো ষ্টেইনিং ইন ব্যাকটেরিয়া মই ফ্লো চাৰ্টটো দিম কিয় দিম মুখস্থ কৰিবা নেকি নকৰিবা খালি কোনটো পইণ্ট ইম্পৰ্টেণ্ট মই কৈ দিম হা ষ্টেইনিং বুলি ক'লে ইট ইজ নট অনলি ওৱান ষ্টেইন দ্যাট ইউ ইউজ ফৰ দ্য প্ৰচেছ we have a mixture of 2 stains that is used for the process. After each stain we wash the bacteria, so question is asked about washing also. Not necessary, always the question is asked about the important point. Staining boli kole what is the stain that is used, hetu question nukhudibo pare because that's very common, right? So certain questions from the chart also or from the flow chart also it has been asked. So we'll write the flow chart. Okay, before writing the flow chart, the first question is why do you stain a bacteria? Because it wants to play holy. Why do you stain a bacteria? Eta bacteria color kyo diyo? Kam nai aru amar no? Exactly, to see the inner contents, because it's a very small microscopic organism. If you want to see the inner contents, then we have to stain it. Similar is the explanation in case of tissues also, right? If you cut a TS of a plant stem or if you cut a TS of a root or a stem, you want to see the anatomy, what you do? You stain it. What is the most common stain that you use there? Saffronin, right? Pinkish in color. Okay. So we stain a bacteria so that, so flow chart kon eta sobore hatot ase niki? Jedi ase I will not write. Jedi nai tetihale likhibo lagibo. Phone ase. So what do you want me to do? You want me to write it down, or it's like you will see it from the PDF? I think that would be better instead of wasting time in writing it down. Online students, do you have the PDF in front of you? Sobore ase no? So staining process tur pora basic questions aha nai. So let's not waste time by writing all the steps, right? What you do in your note, just keep a extra page so that you can write it at home, right? Hetue kor akaron yat likhi thakile samoy bur waste jabo. Okay, just make the heading and keep an extra page for that. Sobore ase ne nai swa? It is page number 59. I think it's page number 59. It's page, or is it again wastage of time? Ako bisariba meliba ako samoy tatke likhiye diyo niki? Likhiye diyo. Tumar nai ta mane? Kyo nai? I know. Hey je mondirot kun khumaise call khwa nai type. I know. Tumar kyo nai? Kyo nai? Where is your mobile? You did not bring. কিন্তু এটা মই লিখি থাকিলে কি হ'ব জানা তোমালোকৰ আছে নাই ফাৰ্স্ট বেঞ্চ একদম বিন্দাস টাইপ ন কোড গ'ল মোবাইলটো কিয় বেগত সোমাই থৈ দিয়া দুখ পাই নেকি উলাবলে হা নেকি কৈছে তোমাক মোক খৰচ নকৰিবা নেকি হে ভগৱান Okay, in 2 minutes, are you all ready with the page or should I write it down? Let's not waste time. Ready with the page? Okay, Jar Jar, ready one, I please share it with someone else. Okay. Okay, talking about gram stain or staining process, the first point that is important is who has developed the process? Christian Gram, right? Very important. Second, what is the stain that is used? The stain that is used is called as gram stain or gram stain to the mono gram stain or crystal violet. So that is another point that you didn't remember. On the basis of staining, bacteria has been divided into how many types? 2 types, gram positive and gram negative. Now we come to the steps, right? If you see the steps, first ki likha ase bacterial smear. Dekha paisa nai? Paisa no? Ebur confusion koratke sidha sidhi likhi diya I am trying to save my time kintu etia aru extra lagise koi thaku no baki bilake buji paise nohole what happens, you know, some other students will say meme okol junkitai paise tahatokhe bujaise muk nai bujua. Don't do that, okay, please. Okay, so we start the staining process. Last bench ase ne tumar te iman dhuniya ke aramot kyo bohi asa question to me usorle aha kabar usorle aha share kora Atke iman 5 minutes ame likhiye dilu heten special request kori pele matibo laiba. Okay, anyway, forget. So staining in bacteria, if you want to stain a bacteria, you have to take the bacteria also. Keneke stain koriba nohole bacteria? First, the bacteria is presented. You have to collect an area or you have to collect a drop of liquid or drop of water where the bacteria is present. You cannot be taking your hand and picking up bacteria like this. Obviously, it's a very small structure. So what we do is, we do something that is called as a smearing process. What is smearing? Bacteria to ami stain koribo lagile, bacteria to ami dhoribo lagibo. Keneke dhoribo? What we do is, we collect the drop of water, we put it in a petri dish or we put it in a slide. To be precise, it will be a glass slide, right? So we take a glass slide. In the glass slide, we put the drop of water. In the drop of water, obviously there are certain types of bacteria, right? The next step that we do is, we take another glass slide and we rub the glass slide on top of this slide, right? Dukhon glass slide long, ekon slide or pani drop to ase, আৰু এখন স্লাইডেৰে এইটোক আমি ৰাব কৰিম এজ ইফ দিছ ইজ এ গ্লাছ স্লাইড উই টেক another slide and we rub it down. This is what is called as smearing. If you rub or slide another glass slide on top of this glass slide, তেতিয়া কি হ'ব এই পোৰ্শনখিনি ইয়াত লাগি যাব না যায় it will get stuck. Is the process understood? Third bench, smearing কি হয় চোৱা এটা কথা কোৱাচোন is the class going very fast?
No ma'am.
ঠিকে আছে বুজি পাইছা ন পিছত বোলে মেমে বোলে পুৰা দৌৰিছে আৰু আমি একো বুজি পোৱা নাই তেনেকা নহয় ন হয় নহয় লাগিলে যি কমকেই পঢ়ো কিন্তু ভালকে পঢ়ো হয়নে লাগিলে এটা লাষ্টৰ লেসন ইম্পৰ্টেন্ট লেসন নহলে মই খালি পিডিএফ দি দিম কিন্তু যোনখিনি পঢ়িছো আমি ভালকে পঢ়িব লাগিব সো স্মিয়াৰিং বুলি ক'লে হু নোজ কোৱেশ্চন আহিব পাৰে ন what is the first stage of staining? Smearing. What is smearing? Glass slide এখন লবা এটা drop of water লবা slide over কৰি দিবা what will be the result? The bacteria will get stuck to the glass slide. That is called as smearing. Next we go for air drying. What is the reason behind? Because there was water, we have to remove the water, so we go for air drying. ঠিক আছে so শুকাই দিলো শুকাই দিয়াৰ পিছত we stain it, we apply the color. The first color that we use is crystal violet, right? Crystal violet is also called as gram stain. It is also called as slash of dia ase so gentian violet, right? All the three names you need to remember. It is called as gentian violet. Once the color is given, the bacteria gets stained, right? Color to jitya di dila te bacteria to nila hoy gol. What next? The next step is you wash it with water. Color diyar pisot you wash it with water. After you wash it with water, you apply another stain. This time what is the stain we are talking about? Iodine solution. So first time crystal violet, step ki monot rakhiba, crystal violet, wash, iodine, right? Since we have applied two types of stain, so they will, after this process they will form a complex that is called as CVI complex. Bacteria to etia duta color thakibo, crystal violet okman thakibo, iodine okman thakibo, duta color jehetu ekeleke thakile we say it is a complex. And what is the name of the complex? CVI complex. So question might be asked, CVI complex form koribo lagile what do? You have to stain the bacteria in two colors. First is always crystal violet, second is the iodine, right? Once the CVI complex is formed, CVI form korile akko ki korim? Wash, right? What is important? A2 wash kihere asile? Water. A2 wash kihere? Alcohol. What next? Once you wash the bacteria with alcohol, wash kora pisot ki hoy either it is water or it is alcohol. Once you wash the bacteria, certain amount of color or the extra color will obviously go away, right? So once you wash it with alcohol, kisuman color gusi jabo, kisuman color thaki jabo, right? After this process we will see that some bacteria get stained and some bacteria got de-stained. So A2 step or pisot আমি কি দেখা পালো যে জিমানবোৰ ব্যাকটেৰিয়া আছিলে কিছুমান ব্যাকটেৰিয়া কালাৰ টু লৈছে কিছুমান কালাৰলেস হৈ আছে, right? So question ahe, what is the step that decides whether a bacteria will retain color or will not retain color? Direct questions aha nai, question ahise kuntu step e decide kore gram positive hobo ne gram negative hobo? It is this step, right? Washing with what makes a bacteria positive or negative? Tumi jodi water koa nohobo kintu. Next question might be washing with alcohol ketia koriba? After crystal violet ne after iodine? This is the way that you have to read about the flow chart, and you don't have to remember the flow chart. Pura direct poribo nalage. ঠিক আছে out of the entire points that is written, this point is very important karon if you wash it with alcohol, that is the deciding factor whether the bacteria is gram positive or it is gram negative. Okay, if the bacteria is gram positive, it will appear purple or blue in color, right? So jinkhini bacteria to color lole tahan to colorful hoy jabo. The rest of the bacteria they were de-stained, no? Why did we color the bacteria? To read about the contents. If a bacteria does not take the color, তেতিয়াহলে tar internal factors pur ami keneke pohim, because after this point we have got two groups of bacteria, one bacteria with color, the other bacteria without color. So the bacteria that had color, it's easy for us to study. The bacteria that is without color, We have to study them also. How will you study? You will go for something that is called as counter stain or restain, right? So, junkhini bacteria color lua nasile, tak ami restain korim. Ebar, what is the stain that you are going to use? Safranin. And that is the reason why gram-negative bacteria appears pink, and that was the reason why we have written the pink color while we were discussing about the cell wall. Is the point clear? So, during the entire process, actually keita stain ami use koru, tinta. Duta talks about, or it indicates whether it will be gram positive or negative. If it is gram negative, then we go for a counter staining process with safranin, which makes the bacteria pink. That's all. Next, from this portion, another question that is asked. Statement to Dibo, gram positive takes in color, gram positive does not take in color. Are the two statements correct? False. Gram positive also takes in color, gram negative also takes in color, but gram positive retains the color, gram negative do not retain the color. So, gram negative a color noloy, this is a wrong statement. Color loy, kintu rakhibo nware. So, we have to use the word retention, right. So, gram positive, gram negative, both of them takes in the color, but as far as retention is concerned, it is only the gram positive that retains the crystal violet or the CVI complex. Clear? That is all about staining. Any points that you did not understand or keep up? Washing to, tomorrow washing to swa, panire wash kori diye slide kon, eko kotha nai. Oh, no na sai, bostu to tat lagi gol no, smear hoi gol, air dry korila hetu to aru stick hoi gol no, right? Jedi tumi air dry nokora, heat disa, heat diya karone tat lagi gol, jedi heat ni diya hi uti jabo. I think it's clear. Kuntu question ahe, kunkini part of our question ahe, we have discussed, right? Next part is we have to talk about forms of bacteria. Talking about shapes of bacteria or forms of bacteria, we always talk about the four types, no? Right? Right. So, apart from the four basic shapes, you have another two shapes, that is a stalk bacteria and a budding bacteria. Why is it important? That two names are important because this is something that is very common, right. Again, when we use the word budding, it generally yeast and hydra that we remember, but it is also associated with bacteria, right. One type of a bacteria that undergoes budding is also called as budding bacteria, okol eta type, that is rotomicrobian. Right. As far as stalk bacteria is concerned, it will have a stalk like structure, and so it is called as stalk bacteria. And out of this four, the most common is obviously Bacillus or the rod shaped, right. Etia, the next part of the topic is you have to talk about the different types of these shapes, right. Coccus or kisuman types ase, Bacillus or kisuman types ase. For example, if you have heard the words called as monococcus, Diplococcus, Monobacillus, Diplobacillus, right? So Streptococcus, you have to know the meaning. Question kya hai? From this section, diagrammatic questions might be asked. For example, eneke diagram di dile and you have to identify which bacteria we are talking about. So since it is round, obviously it is coccus. It has a flagella, it does not has a flagella, it does not has a flagella, that is a characteristic of coccus. Plus 4 of them are stuck to each other forming a group of 4, so that is called as Streptococcus bacteria. So diagrammatic point kini sum, words kita sum. Is the point important? Understood. And examples iman besi important nohoy. Yes, sir. So meaning kita ami buzi pam first. Coccus or are flagellate, right? Because we know bacteria has a flagella and we have already drawn that diagram also. There are certain bacteria that does not have a flagella, and which group we are talking about? The coccus, right? So A2 point of question ahe. Where will I draw? No, দুইটাই Cephalo. মই ভুলকে লিখিছো চাগে। দুইটাই Cephalo. Okay. So the first shape is coccus. What we know, coccus are flagellate and spherical, the first 2 points. Types of coccus, 6 type of coccus. What is important? Examples not important. ঠিক আছে, examples based on iman questions aha nai. Obviously from this part, bacteria part of bahut examples we need to by heart, right? But at which point or under which category we will by heart, we have to be precise. Generally iman bochore ekhini portion of question aha nai. The question that is asked from this portion is obviously diagrammatic based. As I said, diagrams would be given and you have to identify. The second type of a question is a bacteria arranged in a form of chain or a group of aflagellate bacteria arranged in a form of a chain is called as. So you have to know the meaning. So if you are talking about or you are using the word called as strepto, just forget this part. Coccus part to pahori jua, because strepto could be used for streptococcus, streptobacillus, etc., etc. What is important is you need to know the meaning of strepto. Whenever we are talking about strepto, we understand that we are talking about the arrangement. That arrangement is chain lung. So if I say streptobacillus, what you will understand? Bacillus, bahut kita bacillus arranged in the form of a chain, right? If I say okay, I think this is clear. Monococcus, diplococcus is very common. It's 1 and 2. We have a group. Strepto we understood, chain. Next we have tetra. It's also common. It's a group of 4. Tetra mane sarita, we understand. We have a word called as staphylo. Again, staphylococcus. Staphylo, you need to remember, we are talking about the arrangement that is not regular. At a specific number na thake. It is arranged in an irregular bunch. It's not necessary that every time the group will consist of a fixed number. You cannot say that it is a group of 4 members. You cannot say it's a group of 3 members or 10 members. Any member could stay in the formation of an irregular bunch, and at that time we will be using a word called as staphylo. So staphylo ki monot rakhiba, irregular bunch. Strepto ki monot rakhiba, chain. Next we come to this also. Etu upor to question ahise, Sercini, right? Sercini ki, it's a arrangement or it's a group of 8 bacteria, but they will form a cube-like structure. So if the arrangement is like a cube that is made up of 8 aflagellate bacteria, that is called as Sercini. So from this part, what is important? The diagrams and the meanings, not the examples, right? Can I rub the board? Online students, likha hoise? Buji paisa no? Okay. Next likha bacillus. Bacillus heading kori pela first point rod shape, second point most common shape are commonly available, third point flagellate, From this part, first part or ami examples bilak bhalke পঢ়া নাই কারণ আমাক নালাগে এইখিনি, right? From this part অকমান কেতিয়াবা something about bacillus subtilis lactobacillus অকমান question আহিব পাৰে. So the first two examples that we remember. Next two iman important nohoy, but the most important part from this unit is this one, right? So maximum question has been asked up from this word called as matchsticks, right? So whenever you hear this word or question to matchstick dekha pua kuntu arrangement or kotha koi aso, we are talking about pellicid. So certain terms that you need to remember: strepto we understood, staphylo we understood, serchini it's a cube of 8, pellicid just be confined with the word matchstick, baki eko porhibo na lage, thik ase. So matchstick or nisna eta eta ke arrange hote kibo, that arrangement is called as pellicid. লিখিলবা হৈছে নে? Okay, next is the next two types, no? Vibrio eko details na lage. Other points clear? Budhisane? Is it over? Fully? Okay. So we are discussing about the different types of bacteria. Right? We can also say, or we can also talk about this topic as classification of bacteria. যেহেতু different types of bacteria পঢ়ি আছো ন, তো আমি classification of bacteria বুলিও ক'ব পাৰো. So we need to remember that bacteria, if you are talking about classification, we can talk about two types of classification, one on the basis of shapes and the other on the basis of flagella, right? So classification বুলি ক'লে আমি কি কৰিম, bacteria দুভাগত ভাগ কৰিম. Flagella. On the basis of shape, we have talked about the 4 different shapes and the 2 extra. On the basis of flagella, means flagella present ne absent, because we know that there are certain bacteria where the flagella is absent. Together with that, what is the number of flagella they have? Not necessary, each and every bacteria will have one single flagella. In the diagram that we have drawn in the last class, we have shown a diagram of a bacteria with just one flagella that arises from one end of the cell. But bacteria bilakor flagella etatke besiu thakibo pare, and it depends from which point it has originated or where is it present, and accordingly we have certain names for bacteria. Aru eikhini according to flagella yar pora bahut questions ahi jay. So likhason number of flagella buli heading kora. Sorry, number of flagella. If someone has read this portion, can anyone tell me what is the most common question? MCQ point of view, what is the most common question that is asked about number of flagella? Have you heard a word called as peritrichus? Okay, I mean discuss korimi. A bacteria that has flagella that covers the entire body surface. জোৰ তৰ পৰা হ'ব পাৰে গোটেই বডি সাৰফেসটোত যদি ফ্লেজেলা আছে তাক আমি কি বুলি কম পেৰিটাইকাছ এণ্ড আউট অফ দ্য ডিফাৰেণ্ট টাইপছ অফ বেক্টেৰিয়া অন দ্য বেচিছ অফ ফ্লেজেলা মেক্সিমাম কুৱেশ্যন হেজ বিন আস্কড এবাউট পেৰিটাইকাছ আমি আঁকিম এতিয়া again examples not important what is important the terms and the diagram okay when we are talking about flagella we also remember another word that is called as hair like structure of ki boli koy adult plant cellor epidermisor pora kisuman hair like structures arise kore if you are talking about the root or you are talking about the stem we know the epidermis present epidermis ot ki thake hair like structures thake root hair stem hairs for this term we use a term that starts with T ki asile trichome and from that word trichus right so what is trichus that hair like structure are trichus no hair like structure right আর ট্রাইকাসৰ এইটো এক্সাম্পল কিন্তু ৰাখিব ঠিক আছে এইটো বস্তু ইম্পৰ্টেণ্ট মই ডায়াগ্ৰাম আঁকি দিম অলপ পিছত remember the words nitrosomonas and nitrococcus ক'ত আছিলে nitrogen fixation right nitrogen fixation or mineral biological nitrogen fixation or one important question role of nitrosomonas role of nitrococcus no Diagram kita important, diagram kita bhalke saba. If you understand the diagram, we don't have to write it in sentences, right? Kintu yate ami sentence bilak poribo lagibo, and it is on the basis of sentence the question is asked, right? Keneke hude will discuss. First complete this. Over. Arthritis, we understand that it does not have flagella. Why are we drawing this shape? Because we know bacillus is the most common one, right? So we are talking about a rod shape. So let's go for the explanation. Arthritis, we understood. Monotrichus, obviously we understand mono means single. So the question might be asked, a bacteria with a single flagella is called as a single flagella, monotrichus. Amphi mane ki both, right? A bacteria with one flagella at each end is called as amphi, right? Please learn the sentences properly. Next is Cephalotricus. What is Cephalotricus? A bacteria that has a tuft of flagella at one end of the bacteria, that is called as cephalo. It is not one flagella that it has a collection of flagella, but only at one end of the bacteria. Right? So that is called as cephalo. If you are talking about lopho, a bacteria that has tuft of hair on both the ends of the bacteria, many flagella at this end, many flagella at this end also. Lastly, we have the word called as peritrichus, a flagella that has, a bacteria that has flagella distributed all over its body is called as peritrichus. So what is important? Likhi thaba the diagrams and the sentences, not the examples. Examples, eta a flagellator question. And out of all this one is most commonly asked. Peritrichus. Clear? Next heading kora, the structure of flagella. Let me write it down, then we will explain. প্ৰথমে লিখি লোৱা বুজিম right? Structure of a flagella, it has three parts, the basal body, the hook and the filament. I think the diagram itself is understood. Actually this is the portion only that you have to study. Another point that is important is obviously the protein that it is made up of, right? We know flagella is present in prokaryotes, it is also present in certain types of eukaryotes. But one basic difference between a prokaryotic flagella and eukaryotic flagella is flagellin or tubulin protein. Flagellin protein, prokaryotes, tubulin protein, eukaryotes, right? Hetu basi important. As far as these informations are concerned, ekhini okman extra ke information disu. Ketia ba eta question kudi dibo pare, right? One point that is important is if you have, if you remember the diagram that we have drawn in the class, no, prokaryotic cell, or the point of origin of flagella is obviously not the capsule, right? It is from the cell membrane that we have seen that it originated, but we know that a part of it lies in the cell wall also, right? Humai chai bhitorle, cell wall also it pierces and it's present in the cell membrane also. As far as sticking to the cell wall and cell membrane is concerned, the flagella remains attached with the different types of rings that it have. So yate tahator basal part out ketapan ring ase. LPSM not important. What you need to remember, outer pair and inner pair. Why is it called as outer pair? Because it is attached to the cell wall. Inner to the cell wall is the cell membrane. So the rings that is present in the cell membrane is called as the inner pair, right? And the point, gram positive what okal matra eta pair present. Gram negative what two pairs are present. I think that's all for today. This is the content that we will do. We'll continue with the next class, right? Thank you. আজি হল Wednesday no, next আমার কেটে আছে Saturday, Friday no, Friday no, oh Friday to আছে, Friday to morphology hobo, that is in the evening no, next Saturday morning no, Saturday morning ako biological classification, so ratipo ami biological classification gini kori. Thank you.